Okay, concept two notes. We are now going to talk about the cardiovascular system. Some people refer to this as the circulatory system, but I'm going to use language consistent with the Mary B textbook. That is my main reference book. So we're going to refer to it as the cardiovascular system. A little overview. The function of this system is to transport oxygen and nutrients to cells and waste away from cells, and all while maintaining your blood pressure, which is very important. The major structures are your heart, your blood, and the blood vessels that are like the roads or paths throughout your body. And your blood is just the vehicles that are transporting various substances down it. And the heart, it just pumps it all and keeps it all flowing. So again, the heart is the pump, pumps blood through the blood vessels, and your blood vessels form a closed system of delivery routes that begin and end at the heart. Your arteries carry blood away from the heart, so think A for away, and then your veins carry blood to the heart. In pictures, we often show red versus blue to differentiate the two, but in reality, all of them are containing blood, and your blood is red. Fun fact, your cardiovascular system, it's moving approximately 7,500 liters of blood through your heart every day. So that is a lot of pumping and a lot of transport. So learning about the blood and the blood vessels in the heart is so important. We are not going to just talk about it via lecture for hours on end. You're going to discover it for yourself through some discovery stations and labeling the cardiovascular system big body diagram. But for the sake of the video, we're going to keep pressing through. So I'm going to hit some highlights here. The majority of this is going to be 100% review from the discovery station, so it should be very familiar. I just wanted to throw in a few extra things and touch on just a few very important thoughts before moving forward. Tissues of the heart. So the heart is an organ. We know organs are made of tissues and tissues are made of cells. Your heart is composed of strong cardiac muscle tissue. If you remember back all the way to Unit 1, Concept 3, when we talked about histology, we talked about muscle tissue. We talked about it in Unit 2 when we talked about the muscular system. And you have your cardiac muscle, your skeletal muscle, and your smooth muscle. So your heart is made of this cardiac muscle tissue. It has a lot of similarities to skeletal muscle, which we talked a lot about in Unit 2. It is striated, which you can see in the picture, and it contracts using the sliding filament model, which we talked about in Unit 2 as well. But there are some differences. Your cardiac muscles are shorter and they do this kind of branching thing and they're interconnected. So you remember with your skeletal muscles, if we wanted to contract an entire muscle group, we had to coordinate using motor units. Your cardiac muscle, because it's all interconnected, those contractions can be much more coordinated. And that's because there are gap junctions between the cells, which you might remember from Unit 1 when we reviewed cell communication. And these gap junctions allow the cells that make up cardiac muscle tissue to just contract all at once. And that's really important, we're going to see, because we really want your heart to be able to contract and beat in a coordinated and rhythmic fashion. Also, an interesting thing about your heart and the tissues and the cells that make them up are they have very large mitochondria in your cardiac cells to keep them energized. 25 to 35% of your cardiac cells are actually mitochondria. So I think that's pretty interesting. I love making connections between, y'all know what mitochondria are. You've been told since 7th grade it was the powerhouse of the cell. But now we're seeing it in context with cells that are specialized for specific functions, and they have the structures that make that possible. And here's just a small example of that. All right, let's talk more about these cardiac cells that are in your tissues that make up your heart. So 2 types. We have your contractile cardiac muscle cells. These are responsible for the heart's pumping. And then we have your pacemaker cells. These to me are fascinating. They are non-contractile cells, so they don't contract, but they can spontaneously depolarize without neural input. This means that your heart can generate its own electrical impulses to keep it beating regularly outside of the nervous system. So we learned about the nervous system. We learned about signal transmission and electrical impulses and all of that in Unit 3, Control and Coordination. And this is cool to see that in the heart, it can actually do that itself. So how these pacemaker cells work, they trigger their own action potentials. Remember, we abbreviate action potentials as APs. And this is all part of the intrinsic cardiac conduction system. I'm not going to get into a lot of detail on this. I linked a great 9-minute video in the slide notes that you could watch about this. But essentially, your pacemaker cells are grouped in a few areas. The main two are your sinoatrial, the SA node, and the atrioventricular AV node. So your SA node would be like right here. This is your right atrium. It would be referred to as the pacemaker. This is the one that's initiating the original action potential that's going to cause the heart to beat. And then that action potential is received by the atrioventricular node, which is like about right here. And then it gets distributed all the way down to your ventricles and throughout. And there are some fibers and other things that go into it, but this is a big deal. So it starts in the SA, goes to the AV, and then just branches out so that the whole thing is receiving this signal at once. All right, let's review a bit about circulation in your heart. With every heartbeat, the heart is pumping blood through circuits. Basically, there's two main circuits, but you also learned about coronary circulation in the discovery stations. But essentially, the right side, which is over here, remember, because we're thinking about the anatomical position, so it's like within you. The right side takes in oxygen-poor blood from your tissues, okay, and it pumps it through your heart to your lungs, and this is the pulmonary circuit. All right, so oxygen-poor blood in the capillary beds of all your different tissues goes through the heart, gets pumped out of pulmonary arteries to the lungs, and then those lungs fill it with oxygen, pump it through pulmonary veins where it gets received by the heart. Then your left side of the heart takes that oxygen-rich blood that it received from the lungs and is going to pump it out to the rest of your body, which is the systemic circuit. And so these two work together. And so these are in all your different body tissues. So I'm mentioning arteries and veins and capillary beds. You learned about that in the discovery stations, but I'll review those in a minute too when we get to blood vessels. But before that, I want to talk about these chambers that I've been referencing. So your heart has this interventricular septum that divides it laterally into two sides, and those two sides, in total, we end up with 4 chambers. Now, this is the simplest diagram of this I could find. If you get to do the heart dissection in class, you're going to see that it's not these 4 chambers. It's just because of the different papillary muscles and other things that are there, but there are these 4 distinct chambers. So the atria are the receiving chambers. They are the 2 superior chambers, and they are the lower pressure areas. And so fluids move from areas of high pressure to areas of low pressure. So they receive blood from our veins, and because they're moving from an area where it would be higher pressure in our veins to lower pressure here. And they're thin-walled because all they're having to do is pump into ventricles. Okay, so they don't need as much cardiac muscle tissue to do that. Your ventricles, on the other hand, are your discharging or your pumping chambers. These are the 2 inferior chambers. They are high pressure areas so that they can then pump the blood out through your arteries, which would be a lower pressure area. And they are thick-walled, especially this left ventricle. The myocardium, which is the thick layer there of that contracting cardiac muscle tissue, is super thick in the left ventricle, which is something really cool that you'll see in a heart dissection. Okay, so breaking them down one by one, you have your right atrium, which receives oxygen-poor blood from your body, because remember, the right side is our pulmonary circuit. So we're taking in oxygen-poor blood from the body, and then the right ventricle, so it's going to come in here, is going to pump out that oxygen-poor blood out through your lungs to get oxygenated. Then that oxygen-rich blood is going to come into the heart and enter the left atrium. And that's so that oxygen-rich blood comes from the lungs, and then it moves into the left ventricle, which then is going to pump that oxygen-rich blood out through here into the rest of your body. So what are these little gray things here? Those are your valves. There are valves in each chamber to keep the blood only flowing in one direction. We want to keep this path organized. So you have your atrioventricular, your AV valves. These are at the atrial and ventricular junctions. Okay, so they divide the atriums and the atria and the ventricles. They prevent backflow into the atria. So when the ventricles are contracting, we don't want to push blood back into the atria. We want to push blood out of the heart. And so that's what that valve helps to protect against. And then you have your semilunar, your SL valves. These are at the base of your ventricular arteries, so like right here. And they're going to prevent backflow into the ventricles. So when your ventricles contract, we want the blood to go out of these arteries. And so these are going to prevent them from going out and then like sliding back down or something like that. All right, so the tricuspid is the AV valve that has 3 cusps between the right atrium and right ventricle. The pulmonary valve is the SL valve with 3 cusps that guards the base of the pulmonary trunk. The bicuspid or the mitral valve is the AV valve with 2 cusps between left atrium and left ventricle. And then the aortic valve is the SL valve with 3 cusps that guard the base of the aorta. All right, so in class, we're going to pause and we're going to model this pathway because I know I talked about it really simply and quickly, but I get it is complex. And the only way you can really get the pathway down that blood's traveling is to model it over and over. So we're going to do that in class, but keep moving on here. So I mentioned a lot of blood vessels in these last few slides. So just to review, major types, your arteries, which carry blood away from the heart. Your arterioles are mini arteries that branch out of the arteries into capillaries. Your capillaries transfer blood between your veins and arteries, and they're organized into capillary beds, which are just interweaving groups of them. Your veins carry blood towards the heart, and your venules are just your smallest vein components that pull blood out of the capillaries. So all of this is traveling in one direction. So if we look at this little picture here, let's say your heart is up here at the top of the slide. So your arteries are red. That's why I colored it red here. They're sending blood away from the heart and to, a body tissue, let's say in your kidney. Okay, and then that blood's going to go through these arterioles, which are kind of this like peachish color. And then you've got your capillaries and capillary beds, which are the purple. And then once the oxygen is diffused out of it, we've gotten the gas exchange we need, which we'll talk about in concept 3. That oxygen-poor blood's going to go through the venules, which is the light blue, and then the veins, which is the darker blue, and back up to the heart and then the lungs to get oxygenated again. And we're going to pause and talk about some of the major blood vessels in your body through a little activity. But again, for the sake of the notes, I'm going to keep going. So let's talk about your heartbeat. We kind of mentioned it when I was talking to you about the AV and SA nodes, but let's talk about it in a bit more detail. This is what's heard through a stethoscope, like when you go to the doctor. And it's associated with your heart valves closing. So those atrioventricular and semilunar valves closing during something called the cardiac cycle. So it makes a lub sound and a dub sound. Lub dub, lub dub, lub dub. That's what we mean by that. So the lub is when the AV valves, so your mitral and your tricuspid, they're closing when the ventricles are contracting in order to build up pressure that's going to pump your blood out because pressure changes, that's what's moving the fluids. So this would be the point when the pressure in the ventricles is above the pressure in the atrials. And so we have something called ventricular systole or contraction, which begins, which we'll talk a bit more on the next slide. The dub is made when the SL valve, so the aortic and the pulmonary, are closing. And so this happens at the beginning of ventricular diastole, which is the relaxation of your ventricles. All right, so let's talk about, what am I saying? Systole and diastole, what is that? Okay, so your cardiac cycle is the sequence of events in one complete contraction and relaxation of the atria and ventricles of your heart. So systole is referred to contraction and diastole is referred to relaxation. So when your heart is contracting, that's pumping. And when it's relaxing, that's filling. So that's why this picture is labeled heart pumping versus heart filling. And it kind of shows you what it would look like. So just an overview. We have these electrical events, aka your action potentials, that your pacemaker cells initiate. They stimulate mechanical events or pressure changes. So your action potentials, the signals they're sending are contraction, which causes pressure change. Blood flows through the heart, and it is controlled entirely by these pressure changes in your atria and ventricles. So if you're like, why are we talking about pressure? This isn't chemistry. This isn't physics. It's so important in biology too. Okay, so your blood moves down pressure gradients from areas of high pressure to low pressure. So that's essentially what's happening. We're using electrical stimulation, these action potentials, to cause contractions, which create pressure changes, which have these gradients that will then move the blood. Your average heartbeat is 75 times per minute, which would make this whole cycle about 0.8 seconds long, which is kind of crazy. So I'm going to go through it in detail. I don't want this to overwhelm you, but I think it is important to understand. It is a continuous cycle, so it's not something you can really pick, like we're going to start at point 1 and end at, point 2, but I am going to just kind of pick a place to start and go through it from there. So we're going to start with mid to late diastole. So what does that mean? This is a point where you have low pressure in your heart. So blood is just going to passively flow through the atria and open AV valves into your ventricle. So it's just flowing down in there because it moves from high, which would be in your blood vessels, to low, which would be in your atria. Then we have the depolarization, the action potential, okay, that's going to be sent from your pacemaker cells. And it's going to cause or stimulate your atria to contract. And that compresses the blood and increases its pressure. So once the pressure in the atria is higher than the ventricles, blood moves from high to low. These AV valves are going to be forced open and blood's going to be pushed into your ventricles. And that's when we get maximum volume in our ventricles. So 80% of the blood is actually just flowing passively, like what we were talking about up here, from the atria to the ventricles. But that contraction that's stimulated by the depolarization, that pushes that last 20% in, and that's when we get the max volume. So it's going down there. So once that volume of blood leaves, your atria then relax and the pressure in your atria lowers. And we would say this is atrial diastole. All right, then we have the contraction of your ventricles or systole. So your ventricles are then super full. So they're going to contract, which is going to increase their pressure more, and that's going to close these AV valves off. And it's going to cause your SL, your semilunar valves, to be forced open. All right, so what happens when they're forced open? We have ventricular ejection. Your blood is going to rush out the ventricles, okay, through these arteries, the aorta and the pulmonary trunk. And then, because we have that release, we move from high pressure to low pressure. Now the volume's down, your ventricles can relax, which is called early diastole. So your ventricles relax, pressure drops, and then these SL valves will close to keep from backflow back into the ventricles. So what I described here would be, for the systemic circuit, maybe going through the left side of the heart, but the same thing's happening in the right side for pulmonary circulation. It just doesn't need as much pressure because you're only pumping blood just to the lungs, whereas on the left side in systemic circulation, we're pumping blood to the whole body, so it's going to take a bit more force. So despite these differences in pressure, both circuits are still ejecting the same blood volume with each heartbeat, which I find kind of interesting. And we'll talk about kind of what this cycle looks like on an EKG or ECG, an electrocardiogram. That's what, if you've ever watched like any medical TV show ever, you see that when they're in the hospital, it's like the red squiggly line, if you will, the zigzag line, and it flatlines in bad, stressful times in the show. So we'll talk about that in the Exploring Cardiovascular System lab stations. And so we'll kind of make a connection back to this. All right, last thing I want to mention is how this is all regulated. There is so much that goes into this, so I'm just going to touch on some highlights. Yes, the heart generates its own electrical impulses and it contracts on its own, but there are extrinsic things that are controlling it. And you know that because we talked about this before. We talked about this in Unit 3, Control and Coordination, when we talked about the autonomic nervous system, and your body's response to exercise, fright, anxiety. When your body goes into fight or flight, it gets your heart beating faster. So that impacts your cardiac cycle. Hormones like epinephrine that are released in that fight or flight response, and another hormone called thyroxine, which hopefully you remember these from concept 3 of Unit 3. Your ion levels can affect it, your age, your gender, your general cardiovascular fitness and health, and your body temperature. All of these things affect your heart rate and your heartbeat, and thus that affects your blood pressure, which is super important, and we'll also talk about in our lab stations. All right, that's concept 2.